So Leif's group is highly active in the fields of artificial photosynthesis and also proton-coupled electron transfers, and more specifically on fundamental mechanistic studies of molecular systems using a combination of electrochemical, photochemical, and time-result spectroscopic technique. Leif Hammarström is internationally recognized for his leading contribution to these fields, and for many years now, he has been the chairperson of the Swedish Consortium for Artificial Photosynthesis, and is also a member of the Royal Society of Science in Sweden. So Leif, the floor is yours.
Thank you very much, Marielle, for this very kind introduction, and thank you also for inviting me to give a talk. I enjoyed this meeting. It's great to see how a joint grant is fostering or raising an entire community, creating new collaborations that hopefully will lead to more substantially funded individual projects. So, what I've talked about today is mechanism of artificial photosynthesis. So this is Ångström laboratory where I work, and also most of the Swedish Consortium for Artificial Photosynthesis work in this part of the building. We started long ago with building mimics of the electron donor side of photosystem 2 with the chlorophylls, and the first donor is a tyrosine Z. When it's oxidized by the chlorophylls, it transfers a proton to nearby histidine, and later then it takes back an electron from the manganese cluster, which is a water oxidation catalyst. Oops, sorry, I must have. So we made models of this with ruthenium instead of porphyrin, but the proton-coupled events of the tyrosine was there. So I will talk about proton-coupled electron transfer and how important it is for molecular catalysts as well. And I will also talk about some hybrid systems of inorganic materials with molecular catalysts. So this is basically the outline. And I will also eventually go back to some exciting states in exciton diffusion in polymer dots, if time allows. So molecular catalysts. Most of the catalysts that are used today in the field of artificial photosynthesis, the vast majority are heterogeneous. And molecular catalysts have a struggle to keep up with those, but there are some good examples where molecular catalysts have actually been quite successful. And I think in the long run, they have the potential to be much better than the materials. And one of the arguments I have is that especially for more complicated chemistry, like doing CO2 reduction compared to proton reduction to hydrogen, is that we're working with very, very small molecules. And if you look on the CO2 reduction in an enzyme, COD hydrogenase, you see how the CO2 molecule is held together with interaction with two different metal centers and several different ligand interactions. So it's a tight network in 3 dimensions where distances and angles are controlled with distances on a length scale of much less than 1/10 of an Ångström precision. This is very hard to achieve with even with nanomaterials. So for comparison, I just showed the size of a very small nanoparticle. And it's very hard to imagine how you can control length scale that short and also 3-dimensional structure with only materials. But and molecules, of course, you can tune them much more. But if you have many knobs to turn, it's harder to turn them right. But eventually, I hope that we will get there. So enzymes are very good at doing this. They are molecular catalysts. Why are they so much better than the synthetic catalysts that we use, mostly? This we don't know, but we can have some clues. And one of the points that they are doing that is very good, which is important for solar fuels, is the coupling of electrons and protons. Because of course, when you're doing solar fuels, you're making hydrogen or reducing CO2, you need to add electrons and protons together. More than one electron, more than one proton. And oxidizing water, of course, you need to take away several electrons and protons from water. So, PCET, proton-coupled electron transfer, why is it so important? It allows for charge-neutral steps. You don't need to go to potentially high-energy intermediates of single electron transfer or single proton transfer. And you can keep charge neutrality. This is especially important when you are dealing with multi-electron processes, because if you try to accumulate holes or electrons without proton compensation, you're going to build up charge and make it harder and harder to add the next electron and next proton. Very important for catalysis. And of course, you see in nature many examples where you, for example, couple long-distance electron transfer and transport to a proton translocation to generate ATP. This is an example of a copper electrochemistry. We use electrochemical CO2 reduction in proton-coupled electron transfer schemes. You can also do activation of CH and NH-OH bonds in photoredox catalysis by proton-coupled electron transfer instead of the traditional hydrogen atom transfer, and reach much higher effective bond dissociation than you do with the traditional methods, and also different kind of selectivity. So how to describe proton-coupled electron transfer? If the electron and proton are transferred in a single step, you can describe it with a similar, of course, if they transfer in one step, electron first, you have Marcus theory, and you have a similar related theory for proton transfer. Also, if they transfer in a single step, concerted, you have a similar theory like the Marcus electron transfer theory. The idea is that if you transfer charge in solution, you polarize the environment. And if the charge is on the left and then the charge goes on the right, the polarization needs to change from the left side to the right side. And you need to find some compromise in the middle with some semi-polarized state where the energy of the reactant and product states are the same. In a free energy landscape, you draw the famous Marcus curves, the blue one for the reactant and the red one for the product. And when the solvent changes polarization, the energy goes up and down. This is the equilibrium position. And eventually you come to a crossing point where you have equal energy. Isoenergetic reaction is allowed to go into the product. And here the proton is tunneling. And you go down. The difference here is that both electron and proton tunnel at the same transition state. And in the small insets here, you see a different coordinate, which is just a proton tunneling coordinate. The proton needs to go from the left side, from, for example, sitting on an oxygen to going on the nitrogen. And when it's back here, it's not very favorable. And here it's very favorable. And here you have somewhere in the middle where the proton can tunnel. And you see the wave functions indicated here. And it's very important to have a good overlap between the wave functions. So the wave function overlap is critical to determine the rate of proton-coupled electron transfer. And we demonstrated the first series of molecules that probe the distance dependence of proton-coupled electron transfer. So here we have the ruthenium as a sensitizer, which we photooxidized with a laser flash and an acceptor. And then we had different tyrosine units with an internal base linked with different oxygen-nitrogen distances sitting here. And for these four molecules, the distance dependence was huge. So over only 0.2 angstrom, the rate changed by a factor of 100, giving a beta value, which is a slope of this line, of 27 per angstrom. For those of you who are familiar with electron transfer, the beta values are typically 1 per angstrom. So this is about 30 times more distance dependent. So if you're thinking of designing a catalyst, and your rate determining step of your catalysis is a PCET step. And if you're down here, if you then could make a small modification of your catalyst just to go up here, you would gain a factor of 100 in rate of your catalyst without wasting extra energy. This is just the proton tunneling distances. It doesn't require more free energy input. You could do that. You would like to do that, right? Which means that you need to know where you are. Do you have a PCET reaction step? How can I optimize that tunneling? So I've been arguing, as you see here, for 15 years about this thing, that a concerted reaction is really the favorite part for, you should really design your catalyst so that the rate determining step is a concerted, to have low barrier, and you should also make sure that the tunneling distance is good. And this has become quite popular to add these second sphere bases, acid base groups in the vicinity of the metal center to improve catalysis. And, but maybe up till then, maybe we were spending too much time thinking about the electron transfer part of the problem, which is easier, and the proton transfer part is harder. Note that all these effects are ignored in the most common theoretical analysis of solar fuels catalysis. But a factor of 100 cannot be ignored. They are not small effects. One example of a well-designed catalyst is the di-iron center of iron hydrogenase. It has a turnover frequency. It produces up to 10,000 hydrogen molecules per second at very low overpotential, meaning low energy losses. And the active site looks like this. It's a little bit unusual ligands for being a living organism, and it has a nitron here, and I mean, that can be protonated. And when it's protonated and reduced, it's believed the proton is transferred to the iron center, forming this hydride. So this is a critical part of the mechanism. Then it can be protonated again, and here you see then you have a proton and a hydride looking at each other, ready to form a bond. So that's the proposed ideas of the mechanism of hydrogenases. If you look at, there are by now several hundred molecules made, inspired by these sites, that are shown to make hydrogen, but much less efficiently, much less rapidly. They're not at all as good. And one of them is this. Looks very similar. And the question is this nitron, protonated nitron, is it able to form these hydride species? Initially, it could be a terminal hydride, and then swings back to bridging hydrides. We were looking at several of these complexes by time-result infrared spectroscopy. So basically, it's nanosecond laser flash photolysis, but instead of the xenon lamp, we have an infrared quantum cascade laser and an infrared detector to look at the signals. So we can photooxidize the ruthenium complex with a donor in about 100 nanoseconds, and in less than a microsecond, it has transferred the electron to the catalyst, and then we can see what happens. And we can do it in the presence of acid. So what we asked ourselves was, there were groups claiming that this shuttling mechanism of proton forming the hydride was working also in this synthetic complex. The turnover frequency in electrochemical experiments were estimated to be 1,000 to 10,000 per second. So the mechanism would be one electron in, one proton here, then it shuttles to form the hydride, and then you can reduce and protonate again. We thought, okay, can we see this shuttling occurring? We should be able to see this with infrared spectroscopy because of the very nice carbonyl groups. So we did some, with the laser experiments, we couldn't see any evidence. It was too slow. So we had to go to slower timescales and stop flow. And to see, and simply we had an excess cobaltocene as a reductant and some acid. And we could, sure enough, we could generate this singly reduced, singly protonated state. And we were looking at that, and it stayed for a second. And what we actually saw, we think, is turnover going back to the starting state and making hydrogen. And if this is stable on a second without shuttling the proton, it cannot sustain catalysis of 10 to 3 to 10 to 4 per second. So this is direct evidence that this mechanism is not right. This complex does not work with this mechanism. So acid-base groups in the second coordination sphere may look good on paper, but there's no guarantee they're going to work. It's very sensitive with distances, orientations, and energetics, of course. More recently, this was in the summer, we worked on a system that actually does work. So these OH groups in the coordination sphere of a manganese tricarbonyl BIP complex for CO2 reduction. Interestingly, this makes both CO and formate in different. And our postdoc, Samir Chattopadhyay, he could find several intermediates in this mechanistic cycle, including the ones on the pathway going to formate. So he could see the direct formation of manganese hydride followed by CO2 insertion to make the formato complex. And there he found direct evidence that the proton that is used to form that hydride comes from this ligand. So you could see the difference in infrared spectroscopy. So this was about proton tunneling in PCT, the energetic requirements here, and the balance between the different mechanisms, stepwise versus concerted, is very important. So you can go in different ways. Electron transfer first, followed by proton transfer first, followed by electron transfer, or a concerted. And experimentally, it's not necessarily easy. Mostly, it's not easy to decide definitely which pathway it goes. But this cartoon here shows how you go from, in this case, a tryptophan with a base to the tryptophan radical where the proton is transferred. This is a direct concerted pathway. And here you have the stepwise mechanism of electron transfer first or proton transfer first. And you can, this is mainly to show that there are completely different transition states involved. And since this is the lowest state, going from here to here should have the lowest barrier because it uses all the free energy available in a single reaction step, while these are much less driving force and often are uphill. So energetically, this should be favored, but at the same time, it could be that it has the complication of having two particles that need to tunnel at the same time. So it's basically, as I see it, a combination of these two factors. A better lower barrier, classical barrier for the concerted reaction on the one hand, but less complicated tunneling for the stepwise mechanism. And a balance between these factors that decide which way it goes. And this may sound complicated, but if you think about it in simple terms, if you have a very strong oxidant, you tend to go electron transfer first. If you have a very strong base, it tends to go proton transfer first. But if both oxidant and base are weak, then you tend to go concerted. And both oxidant and base being weak means that you have an energy-efficient reaction with low overpotential. So this is what you want. This is one of my arguments why you should design your catalyst to go in a concerted pathway. Unfortunately, almost all molecular catalysts seem to show stepwise mechanism. We were asking some years back, is it possible to actually have a metal complex, a metal hydride being oxidized can we go along the diagonal? Instead of electron-proton in different pathways, can we go along the diagonal, either forming or oxidizing the hydride? This is so here we're not talking about hydrogen atom transfer, but instead separate acceptors and for electron and proton, which is what we have in solar fuels. This had not been shown before for a metal hydride, but the metal hydride, as you saw, is a very important intermediate in catalysis. So what we did was take a tungsten hydride as a model complex and see if we can oxidize it with metal BIP complexes and different pyridine bases. And we changed different iron and ruthenium oxidants and different pyridine bases to change the driving force for the reaction. And long story short is that we could go along all pathways, stepwise electron transfer first, the proton transfer first, or the concerted, depending on the different strengths of these oxidants and bases. So this is an example of what the data looked like. So with a strong oxidant, we see when we don't have the base or add the base, it doesn't matter. The rate constant is the same on the logarithmic scale. So this is clear signature that it's an electron transfer limited mechanism. Just electron transfer and then the proton goes. When we have a weaker oxidant instead, the iron complex, you see a slope here of 1 per 50 millielectron volt. This is exactly what you expect from Marcus' theory for low driving force reaction. This is a clear signature of a concerted reaction. When we have an intermediate oxidant, we see that it still have the same 1 per 50 millielectron volt, but it's faster. This is 150 millivolts stronger oxidant, the gray one compared to the yellow. 150 millivolts correspond to 2.5 pH units. For a concerted reaction, it shouldn't matter if you put extra driving force on the electron or on the proton. You should get the same rate. And indeed, if we shift that line by 2.5 pK units, they line up. This shows a very clear symmetric dependence of the concerted reaction. How can we increase the rate? The rates were not that impressive. Well, without wasting more free energy, we made linked compounds instead to increase the proton tunneling probability. And in some comparisons, we could actually get more than 1,000-fold higher rates. And I'm not going to test you on all the numbers here, but this just basically shows you that the different slopes here show that you have different mechanisms going from the first proton transfer to concerted to electron transfer first as we increase the strength of the oxidant. And this is the same data, but instead of the different pK values, we plot the different pK values there and the different oxidant strengths there, and we see the expected slopes for the different mechanistic regions. This is the first system that where we could show all the three mechanisms for the same system by systematic variation of the driving force. And I think it's to say that this is the best example of a determination of the mechanism, and in this case using free energy dependence. The numbers here are kinetic isotope effects. So we have an inverted isotope effects because of the different zero-point energies of the hydride versus the pyridinium. Here we have a more normal effect that for a concerted reaction, and here essentially no effect because it's limited by electron transfer. So driving force dependence. Marcus theory for electron transfer. That was one of the most important points with the Marcus theory, describing what is reaction coordinate and how the driving force the driving force dependence of the rate constant. And the most controversial part of his theory was that the rate goes up, it levels out, but then it goes down again in the inverted region. And it took 30 years before it was finally experimentally proven and accepted. The inverted region is when the driving force is larger than the reorganizational energy. You see, for a spectroscopist, it means that the potential energy surfaces are nested. So this lies inside that one. So then instead of having a barrier on this side, you get a barrier on the left side instead. And this is very important to keep charge separation in photosynthesis, because you can have a large drive. You can store a lot of energy without just losing it at the same time. You have a lot of free energy, but still a barrier. So this is an important concept. For proton-coupled electron transfer, it was predicted by my collaboration partners, Sharon Hammes-Schiffer, now at Princeton. She predicted that you're probably not going to see this for proton-coupled electron transfer, because you can just put more energy, put the extra energy in the inverted region into vibrations. So if you just put 1 quantum, 2 quanta, 3 quanta into the proton vibration, you're going to have a barrierless recombination anyway, right? And these effects, I mean, you can argue why, but we can talk about it later. But these effects are much more important for a proton than for a heavy nuclei in electron transfer. So together with Jim Mayer's group made this series of molecules, an anthracene that is excited, or a cyanoanthracene, and then it is oxidized in a proton-coupled electron transfer fashion by phenol pyridine. So the phenol is oxidized, the proton goes to the nitrogen. and we have substituents here. So we have 8 different compounds with different driving forces. And we can see that the forward reaction was in the Marcus normal region. And the back reaction for these compounds in 3 different solvents were in the Marcus inverted region, was still concerted reaction. And that showed that under some certain circumstances at least, also PCET can show a Marcus inverted region. For the experts in the audience, we are fully aware that you should not put data in different solvents in the same Marcus plot. This is why this is not a fitted line. It's an indicative region of behavior. So you should, if you should do it more rigorously, we should have multiple curves here. But the solvent dependence itself is an extremely good proof that it is inverted region. It's just a question, you have to just give some idea on how to think about those data in the line. But this is in science, so you cannot go into detail in the plot. So we found this by looking at the femtosecond visible and mid-infrared. It was relatively straightforward. We had a local excited state on the anthracene, lived for on the time scale of 5 to 20 picosecond, depending on which complex we looked at. And that was easily recognized as an anthracene with stimulated emission. Then we had a charge-separated state at a very different lifetime, depending on the solvent and which molecule we had. But it has the very clear signatures here of the anthracene reduced state, with some extra contribution here of phenoxyradical. The cyano group was a nice probe in the mid-infrared, and you bleached the ground state. In the excited state, it shifted out down here, and then when it was reduced, it shifted back a little bit. All consistent. The kinetic traces here show that in dichloromethane, tried 1, 2, 3, showed this behavior. So 1 was the slowest, 3 was the fastest. Even though this had the highest driving force, this had the lowest driving force. So this really shows an inverted region behavior. And when you go to more from polar solvents, the DMF, to dichloromethane, the lifetime of the charge-separated state becomes much longer, because not only does the free energy change, but the reorganization energy goes down, so you get deeper in the inverted region. So this was a very nice result for us. And these molecules showed to have much longer life than we thought. We had several papers on this, and most interestingly, it was a completely unexpected result, complete serendipity. I will not have time to talk about it too much, but it may interest some of the photophysics people in the audience. This is a proton-coupled energy transfer. So when we went to this, this is the, when we go to a rigid medium, for example, of 77 Kelvin glass, the charge-separated state is destabilized, and we open up for a different reaction, where the excited anthracene can transfer its excitation energy to the phenol pyridine in its keto form. The enol form is too high in energy. You can see the blue, this is the absorption spectrum of the enol form, this is the absorption spectrum of the anthracene. You cannot go from here to there at 77 Kelvin in 50 picosecond. It's half a volt uphill. Problem is that you can also not transfer the proton without transferring the energy, because it's also more than 0.15 volts uphill. And consequently, you never see a direct absorption of the keto form. This would be going from here to here, right? So there's no absorption here. So you see anthracene fluorescence here, no absorption, but you see the fluorescence from what we call local electron-proton transfer state, which is a keto form. So this is clearly sensitized by the anthracene in spite of no overlap. So a singlet energy transfer without spectral overlap. But it allows sensitization of dark states thanks to concerted proton transfer. It's a new elementary reaction. I'm good. I have time to even explain. So why did we see the concerted electron transfer here? Concerted PCET in the Marcus inverted region, in spite of the fact that Sharon had predicted it probably not going to be seen. Well, one thing that she did, when she did that prediction, she was thinking about relatively harmonic proton potentials. But if you look on more realistic proton potentials, they look something like this. See, the red one is a double well with a relatively low barrier. The effect is that when you look at the blue curve, which is a short-separated state going back to the ground state, and the real ground state is probably down here. This is now with three quanta, 4, 5, 6, and 7. I see with adding 7 quanta gets you to this state where you have a barrierless recombination. But the 7th state is a very oscillatory wave function. So it just cancels out. The total overlap is 0. And the best compromise is 0 to 3 transition here, where you have some barrier, and you don't have the great overlap, but sufficient overlap. Of course, with the 0-0 transition, you have much better overlap, but then you have a much higher barrier. And these effects are there because you have this very anharmonic potential. And it turns out that in Sharon's calculations, many systems actually have much more anharmonic potential than we usually think. And this is the explanation why we saw the inverted region in spite of her earlier prediction. This is not necessarily universal. We are looking to try to design a system that doesn't show the inverted region with more harmonic potential. We'll see if we manage. Okay, enough of PCET. Now, let's talk about quantum hybrid systems. So this is a more detailed story. It's basically a single paper story. First this and then another one on polymer dots. So this was a collaboration with Muriel and Vincent Artero. And we had already started working with copper indium sulfide quantum dots. They're quite tolerant to off-stoichiometric ratios between copper and indium. And we're interested in pushing up the conduction band a little bit with low stoichiometry copper. And they are made by, we can make them in aqueous synthesis, which is nice, benign environmentally. They are quite reducing. And we didn't, the only passivation we had no shell here, but there was just L-cysteine and iodide. And then we're interested to see, could we have a molecular catalyst on this surface and see electron transfer? And could we actually demonstrate that the catalyst is reduced? Because up to that time in the field, no one had shown that. They assumed that if the quantum dot is quenched, it must be electron transfer to the catalyst. There are many reasons why things can be quenched. So I think we thought that it could be a good thing to show. And if you look at the absorption spectra here in black, you see not a very distinct spectrum, but this is typical for most quantum dots. And you see photoluminescence here in the red. So the band gap here is around 2.4 electron volts, 520 nanometers. So with these quantum dots, we could actually show the signature of the reduced catalyst here by the infrared absorptions of the carbonyls. But then in collaboration here, we worked instead of a better catalyst, which is a cobalt catalyst that they developed in Grenoble. And when we did the photocatalytic experiments here, we could see that we got quite decent hydrogen generation, and over 8,000 turnovers per catalyst. So they were quite robust. So this is 24 hours irradiation. What is interesting here is that we use 2 different catalysts, one with a designed anchoring group, one without that anchoring group, and they show essentially the same results. This surprised us. So one is attached, one we thought was not attached, but they work the same. Another thing which is interesting is, of course, that with just 1 micromolar, we get about the same result as with 10 micromolar. So adding more catalysts doesn't really help in this range. So 1 micromolar seems to be sufficient to take care of the reducing equivalents. And then, of course, that means that the total number per catalyst is best for the low catalyst concentration. We look then first to characterize the binding here by fluorescence spectroscopy. So we're looking at the quantum dot fluorescence, and this is with different concentrations of the quantum dots and the two different catalysts here. And we see this upward curvature, which is exponential. So if you take the logarithm of that fluorescence intensity ratio, we get very nice straight lines up to quite large numbers. So this can be explained by a Poisson distribution of quenches over quantum dot particles. So it's enough with 1 catalyst to totally quench the quantum dot. In the lifetime measurements, we saw no difference in fluorescence lifetime, because then we only see the ones that don't have a quencher. If you assume a Poisson distribution, then the probability that the quantum dot has no quencher decreases exponentially with the average number of quenchers per quantum dot. That means that the fluorescence ratio F0 over F increases exponentially. This fits very well with the data, and if we normalize for the quantum dot concentration, here we get essentially the same slopes. Here we get a little bit more variation, but it shows that even with about 1 micromolar catalyst and a little bit less than 1 micromolar quantum dots, we get almost essentially complete binding, even with a compound with no binding group. So they really like each other. And this is interesting, because usually most people say that the limiting factor in such systems is the electron transfer from quantum dot to catalyst, because they need to diffuse together. That doesn't seem to be right here. It's clearly not right here. In transient absorption spectroscopy, we could also see direct evidence. So this is about the average number of quenchers is 0.7. So here we see in the mid-infrared, we see in blue without catalysts, and green and red the 2 different catalysts. They're indistinguishable, but they're clearly faster. We see the electrons in the conduction band disappear faster than presumably because of they reduce the catalyst. The ground state bleach recovery see a similar increase in rate, and they end up in a positive absorption. And if you look after 2 nanoseconds at the spectra, you'll see that there is still a bleach here without the catalyst, but with the catalyst you see an increased absorption here, which we know from the reference measurement of spectral photochemistry is a reduced catalyst. So direct observation of the reduced catalyst. So it's clear that the change in dynamics is really because of electron transfer to the catalyst. And this is on a time scale of average lifetime of about 10 picoseconds for the electron transfer. So I didn't talk about that, but we assigned the different bands in the transient absorption of the Q dots. We have a strong binding of very different molecules. We tried with molecules with opposite charge. They bind. We tried molecules with no charge. They bind. Not exactly the same, but similar-ish. So if you try ferricyanide, methyl viologen, and an uncharged iron complex, and they all bind, we cannot blame it on electrostatics. And I'm a physical chemist, so what do I know? But I can't really see the chemical common denominator between these 3, except that they're able to quench the quantum dots. So I don't understand the nature of this binding, which is interesting, but it works in our favor. We have direct evidence for ultrafast electron transfer catalyst, and this is not rate limiting for photocatalysis in these systems. And the photocatalytic performance was comparable to those that have a zinc sulfide shell to protect from charge recombination. So we can, of course, debate if this is which one is better, but they were surprisingly good. Finally, a little story on exciton diffusion in polymer dots. First, what is polymer dots? You know about organic semiconductors. They're used in solar cells, for example, and for the photocatalysis. The good point of making them in small dots, nanodots, less than 100 nanometers, is that you have a shorter diffusion length that is needed to get from where you excite it to the surface where catalysis happens. So you have a better chance of doing charge separation at the surface than if you have a bulk material or thin film. In general, they are interesting materials. These P dots, they're easily to tune, easy to synthesize. You can modify them. Relatively good electron and hole mobility. And my colleague here in Uppsala, Hein Yang, he was the first to show proton reduction many years ago with his P dots, that he called them. Okay, they're also biocompatible, but that's not my talk. So the question here, if you now form an exciton somewhere in the polymer dot, they can recombine in the bulk, they can recombine at the surface, but they can also dissociate at the surface. And the question is, what is the fraction, how many of these excitons are actually reaching the surface where they can do useful chemistry? There were two previous studies of the PFBT molecule, which is a common polymer. We were not very happy with those. It only had fluorescence intensity data. Some of this was very confusing. For the aficionados can look at this plot and tell me how the hell that is supposed to make sense. And they didn't agree with each other. There were also some large defects. So we thought that can we do better? We took the PFBT polymers together with a polystyrene PEG to stabilize it. So PEG stabilizers with a hydrophobic core. And then we doped it with small amounts of perylene red as quencher. So PFBT perylene red has a first radius of about 3 nanometers. And then we're looking at both steady state and time resolved fluorescence to see if we can get a consistent picture from both experiments. And we did analytical fits instead of computer simulations of the dynamics to fit the data. So the hydrodynamic radius is 50 nanometers from DLS. The core, the hydrophobic core, is less than 10 nanometers based on the relative masses that we know. For 1-to-1 ratio of these two is about 8.5, I think. And when we have 3 times this and 1 times that, the core is a little bit smaller than 8.5. Okay. In the absorption spectrum, you see PFBT, and when you add small amounts of the quencher, you have to squint to see the absorption of the quencher, but it's here. But in the fluorescence, it's very clear that you quench the donor fluorescence, and instead you get the fluorescence from the acceptor. But if you look somewhere here in the middle, you continue to quench the donor, but you don't get more fluorescence intensity from the acceptor. So here we think that if you go more than 15 nanomolar or something like that, we start getting aggregation, the interaction between the perylene reds. So it turned out we avoided that region. And here we assume a Poisson distribution of quenchers. The difference in the previous case is that we cannot assume that the fluorescence is entirely quenched just because they are in the same volume. Instead, we have to use the we choose to use the Infeld-Attachia model that was developed for fluorescence quenching in mice cells. It assumes a Poisson distribution of quenchers over mice cells, and the lifetimes or the time traces are fitted by this equation where you have the decay rate constant without quencher, average number of quenches per micelle, or in this case, per volume that the exciton has time to explore. You have the quenching rate constant for one one quencher in the volume. And this is just to fit without quencher. And we had the bi-exponential. We needed 2 exponents to fit the data. So we pragmatically just said, OK, let's take 2 of these terms and add them together, one with this lifetime, one with that lifetime. We assume that the KQ is the same. These just reflect a slight heterogeneity in the environment. This fitted very well, data. What is different from the case with my cells is that the core volume can be larger than the exciton diffusion volume. In my cells, it's clear that it has time to explore the entire micelles. But what if the exciton cannot diffuses too slowly, so it actually decays before it reaches the surface? Then the volume that we get out from this occupancy will be smaller than the physical volume of the core. So the results here are simply like this, with 3 different concentrations. With 1-to-1 PFPT and PEG, we get very good correspondence of the occupancy and the relative concentration, and a quenching rate constant of about 0.5 per nanosecond. When we have the somewhat smaller cores, because we have more of the PEG polymer, we get about twice the quenching rate constant, which makes sense because the core is smaller. It's going to quench faster. And a little bit less good correspondence here, but it's OK. And then we get quenching volumes in the 2 different cases of 2,300 cubic nanometers. This is equivalent to 8.2 and 6.6 nanometer spheres. This fits within all experimental error of our estimate of how large our cores are. We think that these volumes are actually limited by the cores. So good. This is a very efficient exciton diffusion in the entire core. So, but what is then how can we compare this with measured exciton diffusion lengths? Exciton diffusion length is the root mean square displacement, typical textbook definition. But the question is, if you have a root mean square displacement of, let's say, 8 nanometers, it doesn't mean necessarily that you explore an entire ball of 8 nanometers. What's the relation here between these two? Look here, this is a random walk in 3 dimensions. If I don't know exactly where this is supposed to start, but let's say that this starts here and ends there. This would be the root mean square displacement. And you clearly see that most of this area is void. It hasn't been there. This is another picture. If you want to see, for this random walk, how large volume has it explored, then you have to draw, then you see the particle moving here. And in our case, the thickness of that tube is going to be determined by the first radius. If there is a quencher nearby and the first radius is 3 nanometers, then it's going to be quenched. Otherwise, it's not. So then it explores this volume. What is that volume for a root mean square displacement of a certain number? It sounded to me like a difficult problem, interesting problem. Someone must have solved this in a completely different context than polymer dots. I looked for a long time. I didn't use the right keywords. I can tell you what keywords I should have used, but you will see it. It's a Wiener sausage volume. Because these are called Wiener trajectories. And if you then put the volume here, this is your Wiener sausage. Ha, ha, ha. How funny they can be. Statistical physics. Our student loved it, of course. So this is basically the thickness of the sausage here is the slices here are determined by the first radius. So this has been apparently a problem in statistical physics since the 1930s, and this is a very nice paper. I could understand the important parts in Journal of Statistical Physics. Amazing. And from the equations they presented for 3-dimensional, the relation between volume and the mean square displacement is this, 2.1 times the first radius. It means that we get an exciton diffusion length of 90 nanometers, which is about 1.5 and 2 times longer than was reported in the previous studies. And I think, we still have to remember that it's limited by, if our polymer dots would be larger, the volumes may be larger, and maybe the real exciton diffusion length is even longer. So, we used an analytical solution to time-resolve quenching data and got satisfactory fits. And it seems that the exciton are able to explore the entire core volume. Much longer diffusion lengths suggested, which means that, the bottom line, the exciton should have a high probability to reach the surface, which is important for photocatalytic applications. And with that, I'd like to thank all the people involved at Uppsala, my senior colleagues in Uppsala for different things. Vincent Muriel at Grenoble, Princeton, Sharon Hans-Schiffer and a group, Yale, Jim Mayer and his group, the people who gave us money, and you for your attention. And a shameless advertising. I'm associated for ACS Artificial Photosynthesis, a new journal, which is in collaboration with Westlake and Dalian Institute of Chemical Physics. So the editor-in-chief, and we are the 3 associate editors. We accept all high-quality manuscripts in all areas related to artificial photosynthesis. So I encourage you to consider our journal for your next submission. Thank you.
Thank you, Leif. So we have time for some questions.
So thank you very much, Leif, for a really wonderful lecture. Enjoyed it very much. I wanted to go back to the inverted region for the proton-coupled electron transfer that you showed us. You really convincingly demonstrated how important the reorganization energy was in having this proton-coupled event. And then I noticed that you actually chose solvents that had very low reorganization energies because they were non-polar solvents like dichloromethane. And so I was wondering if in this case you weren't decoupling the electron transfer and the proton transfer, which kind of brought you back a little bit more to something similar to the intramolecular inverted region seen by Gerhard Close and Miller.
It's an excellent comment. So we have done this all from DMF down to toluene. And of course, what happens when you go to too low polarity solvent, we start destabilizing the charge separated states. So then we have this proton-coupled energy transfer competing. But in these solvents, we didn't see that. It's still a concerted reaction. You may think that dichloromethane is, it's actually quite polar. So it's not, you still have a large polarization of the solvent because of the charge transfer. Then, if you go to the classical Marcus theory based on dielectric continuum, you may wonder what happens in toluene. In principle, toluene or alkane should have zero reorganization energy. And this has been shown by lots of theoreticians that this is not the case. The reorganization is much higher than the continuum model says. I don't have a personal opinion on that from our data, but I think this would certainly apply also for a PCT reaction. I think the reorganization energy is larger than at least in many cases.
All right, thank you.
Mm-hmm.
Other question?
Thanks, Leif, for the talk. It's interesting that you started with something molecular and then you ended up with something material. And I think my visualization got lost now with the P dots because of course you first talk about this exciton and forming let's say, the charge on the surface, but now I miss the charge accumulation needed to do, let's say, proton reduction. So I don't know if you can comment on how these things would change now that you need to accumulate 2 electrons and 2 protons, for example, to do hydrogen evolution.
Yeah, no, so the transition from molecules to more materials probably reflects my neurodegeneration, but, but seriously, obviously here we are looking at an isolated part of the problem, just the excited states. So one can discuss, are we really accumulating charge in the p dot? We think that we don't really have separate charges. We have excitons in the p dots. Do we have 2 excitons at the same time under solar radiation? Probably not. They have too short lifetime. So the accumulation of charge has to happen here at the catalyst, which we didn't study in this paper. We have, of course, studied in other papers, but if we have a platinum particle there, it's hard to count the electrons. But for sure, the systems as such work for proton reduction and CO2 reduction. My colleague Heining has shown that. But for the accumulation of charges, we have to rely on the catalyst part.
Okay. And does it also include the part where you now oxidize something to have the complete one, or it's sacrificial source for this?
Yeah, in his experiment on photocatalysis, they have been sacrificial, but he has managed to get some hydrogen peroxide. So not a good system yet that combines the two, but has separate half reactions. And Kevin Sivela has some very nice work where he actually uses organic material for water oxidation. Sounds like a suicide thing, because they're usually easily oxidized, but he's shown that with the right tricks, you can get them to last quite well.
Thanks.
Thank you. Any other question? Yes.
Yes, sorry. Thank you very much for your beautiful talk, and so giving a lot of fundamental aspects on photophysics. And I have a naive question regarding also this proton-coupled electron transfer. In my mind, I thought that proton transfer could be also driven by tunneling effect, and so very quick, I would say, process. And so I was wondering to which extent here you are not considering, for instance, water, where also it can drive at a very long distance some proton transfer. So my first question is how tunneling effect can be taken into account, to which extent also water can also help, this proton transfer. And finally, are you tried also to investigate this proton transfer in deuterated or with deuterated molecules?
Yeah, sorry, I was trying to get back to that.
Sorry.
No, you're absolutely right. The proton is tunneling here, but of course the tunneling step itself is very fast. But when it tunnels, but there's a probability which depends on distance. So when I said that, so with this rate dependence on proton tunneling distance, this is a probability. It's not a slow migration of the proton. It's really one-step tunneling in all cases. We have shown this in some cases. We have some small synthetic proteins, for example, where we have shown that the buffer is not the proton acceptor; it's water. So we can, by simply varying the concentration, for example, we can see that it must be water that is an immediate proton acceptor and so on. So yes, you're absolutely right there that water can be very important. In some of these systems that I showed with the inverted region, for example, we didn't have water. But in these small proteins, we have some intricate effects of water there because there we have a tyrosine or tryptophan buried with very little solvent exposure, and still it seems that they are oxidized happily. So there we think that there's we have some indication that there's some partial unfolding of a chain that allows one water molecule to come in and act like a proton bridge, if you want. Maybe this is similar to what you... So absolutely, you're right.
Thank you, Leif. So, thank you for this very nice talk.